Welcome to the BRC Statistics Hub Lunchtime Seminar Series. Today we are talking about good practice for statistics for clinical trials. My name is Sofia Massa and I'm a lead statistician in Octro in Endorms. This is the plan of the talk. First of all, we will look how statistics underpins all aspects of clinical trials. Then we will go through some potential bias consideration arising in clinical trials, some statistical input that is required at each stage of randomized controlled trial, some details about the statistical analysis plan and why it is important and the content of a statistical analysis plan, some data quality consideration, statistical programming and coding consideration, and finally, a good practice requirement for statistician and for reproducibility of research consideration. This diagram shows how statistics is at the center of all the important stages of a clinical trial. In particular, it is related to the design of the clinical trial itself, to the methods that are going to be used, to the data and the design of the data collection system that are going to be used, to the analysis of the data collected, and finally to the interpretation of results. Statistics is connected to all of these stages, but also all of these stages are interconnected between them. So statisticians play a crucial role in clinical trials research, and the key role of statistician is now recognized also by regulatory agency and funding bodies. Let's have a look at what are the potential biases in randomized controlled trials. First of all, the definition of bias. Bias is the deviation of results from the truth due to systematic errors in the research methodology. Selection bias is due to the methods used to assign patients to study treatment groups. Performance bias occurs when patient or clinician are aware of the assigned treatment and perform differently as a result. Attrition bias is due to an influencing factor that can cause non-random withdrawal from the study groups. Detection bias is bias in the measurement of study outcome when outcome assessors are aware of the assigned treatment. And finally, reporting bias is bias in the outcomes reported by a study, mainly when non-significant findings are ignored. How can we minimize or avoid the biases that we have just introduced for randomized controlled trials? For selection bias, we can think about random concealment and therefore using randomization to allocate participants to the different groups. For performance bias, we can think about blinding and therefore allocating the participant to group without them knowing to which group they've been allocated. For attrition bias, we can monitor the withdrawals rate in the two arms of the trial, or for example, give incentive to the participant for them to stay in the trial. For detection bias, we can think about performing blinded assessment, so the clinicians are blinded to the randomized group in which the participants are. And finally, for reporting bias is to pre-specify the analysis before having finished collecting the data and starting the analysis. As we said at the beginning of this talk, statistics is crucial for all the stages of a randomized controlled trial. What are the questions that can arise in all the possible stages? For example, at the design stage, we really want to formulate models that address the question of interest, and they're not only of interest for our statistical considerations. What are the sample size assumptions that are important to know in order to do the correct sample size calculation? Which data do we need to collect? Are we sure we are not collecting too much data or too less data to answer the question of interest? What are the randomization choices and what is the appropriate method of the randomization to be used with a particular design? For the data collection and analysis, it's paramount that we collect excellent data, because if we don't collect excellent data, we are only going to have a lot of problems when we start analyzing it. Therefore, we want to collect data that is as accurate as possible, that have very few missing data, that has not been contaminated, or there are unappreciated errors. The coding for the analysis is also important, so preparing all the code that is needed to run the analysis when the data collection is finally ended. And the further question when the analysis is undertaken is what are the level of uncertainty involved in the conclusion of the statistical analysis? Do the results change if we change the assumption? Are sensitivity analysis correctly performed? And when we report, it's very important that the reporting is not biased in any way. So then we either undermine the results or overestimate the results that we are reporting. Let's have a look in more detail at the design stage. First of all, it needs to be extremely clear what is the research question being investigated. So good discussion with the PI, with the CI, with the clinical people that are sort of defining the clinical question are very important. Which treatment effect do we aim to quantify? Both answers to these questions impact the choice of the study design, of the statistical method going to be used, of the data to be collected. The statistical methods needs also to be defined at the design stage because we said before that they need to be defined in advance. So we need to define them within the estimate framework. We need to decide on the sensitivity analysis, for example, or other important analyses. What are the exploratory analysis and what are the primary analysis, depending on what are the primary outcomes, what are the secondary outcome, and so on. Also, we really need to ask, do the statistical analyses that have been chosen, they really answer the clinical relevant question? Or again, it's because we want to use the latest methods, but perhaps they are really partially important to what we are doing. And then again, at the design stage is very important, this data collection planning. This is essential to determine what can be estimated, how reliably can we estimate it with the data that we collect, and also what data are required to support estimation of the treatment effect, which data can be collected, do we need different type of sources of data, for example, coming from public registry or coming only from trial data or coming from lab data, where do we collect all this information, how we integrate it all together so that it can be used for the analysis. Let's move on now to the statistical analysis plan. As we said, it's important that the statistical conduct of the trial is well documented, is excellent, and is transparent. And we have to think about what is the influence of every statistical decision on the trial conclusion. In order to do this and to predefine all the analysis, everything should be documented and defined in advance in the statistical analysis plan. It's not a standalone document and should always be read in conjunction with the clinical trial protocol where all the other procedures are described. Also, when the analysis is starting, the SAP needs to be followed, but when we have a clean and validated data set for the analysis, there is no point in having a well-documented SAP, well-refined, but then the analysis is performed to a data set that has not been clean, has not been validated with a lot of inconsistency. This will not bring good results to the trial. It's also good to bear in mind that some journals now are requiring the SAP to be submitted alongside the report of a clinical trial, so then it can be used in the peer review process. It can be also made available as a supplementary material or published as a standalone article, so it's good that it's of a very high standard. There are also some guidelines that have been published for the content of statistical analysis plans in clinical trials. In the published guidelines, there are specific items that need to be addressed in a clinical trial SAP. This is an example reported here, but there are specified all the sections that need to be reported, all the items that need to be described. And therefore, it's sort of easier to prepare the SAP because we know all the steps that need to be followed and all the sections that needs to be addressed. There has been recently quite a few initiatives that have increased the quality of the sub-content, and some of these are the estimates, so the clear definition of what is the estimates of the trial, of sensitivity analysis, initiative regarding data sharing, so how we can make sure that the data that we produced can then be shared, with mandatory requirement to post clinical trial results in publicly available data set. So there are very important articles that have appeared recently, and there is a list here, but it's not exhaustive, with reference pointers to the estimate frameworks, to how to address them, and why it is important also to share trial data. Let's have a few considerations now about the data quality and the data completeness. Again, in order to set up data collection system and decide on the data to be collected, there needs to be quite a strong communication with the IT team available in the clinical trials unit, for example, because the way the data are defined, collected, and handled impacts the quality, which in turn affect data use. So this diagram here, it really shows that how the data is observed, how the data is processed, how the data is represented as an effect on data quality, but the data quality, of course, impacts the data use, and the data use impacts the data quality. So we have sort of a feedback loop. So it is again paramount that we collect excellent data, as we've already said it before, and I will never stop to repeat this, because otherwise we are not able to draw reliable conclusion from the trial data. And we need to keep in mind that setting up a trial, designing a trial, sometimes can be quite a long process and quite intensive in terms of resources needed. And it would be a shame if at the end the results are not reliable because of data quality and data completeness issue. There is this nice paper mentioned here from where the diagram is taken that has very important consideration on data quality issues. Another important component of statistical good practice is the statistical programming and decoding. As we've said before, data quality, planning for data collection is very important, but when we have the data, when everything has been planned very well, then we need to make sure that also the statistical programming is done to a very, very high standard, therefore including checks, validation, and so on. The statistician might be involved in all of the following steps, usually for a randomized control trial. So for example, setting up the randomization, so the programs to generate the randomization schedule, data exploration to explore the nuances in the data but without any comparative statistical analysis. When complex checks are done, for example, to derive primary outcome, to derive data sets or identify membership of the analysis sets. When statistical monitoring is needed for producing reports for TSC meetings or DSMC meetings and so on. And finally, for producing the result for the interim and final analysis. So all of these parts are important, but essentially every unit should have their own validation checks, statistical programming rules in order to make sure that the code use is reproducible, that the results are reproducible, and all the results essentially are consistent with the data that was given and so on. Another component of statistical good practice is reporting. So when the study has been designed, the data has been collected, the statistical program has been all in place, and finally the analysis has been conducted, it's very important that the results are reported following guidelines such as spirits and consort, because these are evidence-based guidelines. They comprise a checklist of essential items that should be included. The spirit is for the protocols and the consort is for the primary reports of completed randomized controlled trials. They provide guidance to the authors on the minimum information that should be included in the reporting of trials, so that essentially we make sure that all the reports and the protocols are clear and transparent, and any person that is going to read the report is able to find all the important required information to understand the full picture that the results provide. There has been an update very recently on the consort, so there is the consort 2025 statement, and all the literature essentially can be found within this article. As we said at the beginning of the talk, the contribution of the statistician to the design and analysis of a clinical trial is essential. But we need also to be able to reconstruct and to find all the statistical contribution to the trial. And of course, this requires a rigorous and transparent documentation that is evidenced in the reproducibility of results. Of course, a lot of things can happen during a trial, during a randomized control trial, and it is important that we consider also how long does it take to reproduce all the results, what are the results available, do we know everything that has happened to the trial, do we know all the history of the trial, anything that has, for example, happened to the data collection, is everything recorded? Because one thing is just to be able to apply some programming and being able to reproduce the results, but we need to have all the context in place, all the information about what has happened in the story of the trial. From a statistical point of view, it is now considered best practice to generate a statistical report directly from statistical programming, because in this way we remove the possibility of introducing transcription errors, and also we can reconstruct the analysis in a simplified way. There is also a paper pointed here in determining a risk proportional approach to the validation of statistical programming for clinical trials. And it's important to note that a risk proportional approach is needed because we need to have a balance about sort of how long will it take to recreate all the analysis, what is the risk involved with any decision that we take. A few extra comments on the reproducibility of results. As we said, we want that every piece of analysis can be tracked back to its origins in the raw data and in every derived data set, and everything essentially can be found from the input to the code to the output. There is some developments in best practice for statistical computing. I've added here 2 possible articles, but there are many more appearing now, and on essentially rules for digital data storage and so on. We have also to keep in mind that it's becoming very common now for having data sharing agreement and to have requests for data sharing of data that has been used in randomized clinical trials. Of course, after all the consent and all the regulations sort of are respected, it's very possible that we are asked to share anonymized or pseudo-anonymized data set, and therefore even more people will look at analysis, will try to reproduce it, maybe to compare methods and so on. So we need to make sure that all these rules, all this best practice for statistical computing, for the code, for the derivation of the outcome, and for all the results that appear in the statistical report, essentially, they can be reproduced. I want to conclude now by saying that all the topics that we have covered for good statistical practice are now included in a good statistical practice training for statistician. This has been developed a year ago because it was noted that mandatory requirement for good practice training was required, but there wasn't anything specifically related to statistician. So a working group essentially has developed a role-specific, a comprehensive, accessible training which is tailored to statisticians that are working in clinical research, and particularly in the clinical trials arena. And this encourages best practice, leading to transparent and reproducible statistical activity, as it is also required by regulatory authorities and funders. So I really recommend reading this paper and all the references therein in order to see what is required now to statisticians working in clinical trials. Thank you for attending the talk today. Do you have any questions?